thank you, organizer, for give us this opportunity to share our research with you. And first, why it matters to detect selection pressure? The short answer is to understand the evolution. Let's start from this long journey from the Darwin. So Darwin's basically suggesting two types of selection and leading to explain the origin of species. The first type is the native selection, which corresponding to the disadvantage mutation, disadvantage kind of change in the genetic material, and it will be purified. The second type is the advantage adaptive mutation change that will be eventually fixed in the population. So he's suggesting that two types of the selection and two types of the mutation, okay? And we know this is a survival fittest, so that basically means the environment keep changing, so we expect the response will keep changing. Okay, and 50 years, roughly 50 years ago, Kimura adding the new type of selection, which are the genetic drifting, neutral selection, basically no selection at all. And then this can help to explain some of the molecular clock observed from the data. So, but this is creates a big debate. For the fixing mutation, fixing change, are they from the Darwin adaptive selection or they from the neutral selection? Okay, so this debate really started 50 years ago and has not been ended. Okay, that basically means for our genome, we ask, where they are from? Are they from the adaptive mutation or they just from the genetic drifting mostly? And so this is actually very nice kind of the review on this topic, and one defines the more kind of the terminology for resolving this kind of debate. And I argue to ending this debate, we need very good test for the selection pressure so that we can classify the mutation based on the fitness change into the advantage mutation, disadvantage mutation, or the neutral mutation. Maybe you have something between. Okay, so added to the content of the molecular biology, we know the genetic information stored in the nucleic acid, mostly DNA and RNA, and they can transcribe into the transcripts, and transcripts translate into protein. But we know that oversimplifies the complexity. We do have some uncoding region, like here show a vowel, and you have the uncoding region we call the UTR, untranslated region, and they also can play some important role in regulate gene expression. But nonetheless, you have the genetic information, so the phylogenetic tree can be inferred, and then put a very simple aspect of the two type of the evolution mechanism. You have the most mutation, most change are disadvantage. So they're going to be removed out from the evolution. But then you have something are neutral, okay? Even slightly deleterious, they can also kept and fixed in the population by the genetic drifting, but you have some the advantage mutation that leading to the speciation, leading to the evolution to more complicated organism. Okay, so we want to put this content and to detect that kind of selection pressure. So KKS, this is one of the common tasks for doing that. It's wonderful, and the other name for this is DNDS. And so how many already use this for detection? Okay, I saw Many has used that. Okay, it's a wonderful tool. However, this require the strong assumption. The assumption is our reference state. Reference state is KS, which are synonymous mutation. So based on our dogma of molecular biology, it basically means, well, synonymous means does not change protein sequence, does not change protein structure, so they have no consequence on the fitness change. Okay, so if that's true, then the non-synonymous mutation, which opposite, change protein sequence, change protein structure, and more under the selection pressure. So we can use that to compare our reference. So that's Ka-Ks ratio. So if we saw the non-synonymous mutation rate higher than the synonymous one, so that basically means it has an advantage, and it's fixing faster in the population. So we call the adaptive positive mutation. That's the Ka-Ks greater than 1, right? And if those two rates are the same, there's no selection. That's neutral. And opposite, if you saw purified selection, then it will remove. Well, all what I said is depend on you assume synonymous mutation are neutral. Is this true? Well, that's a question mark. If not true, then your K-Ks ratio will not able to provide that information. So that's what we're tacking on it. And then in the uncoding region, you know by definition it's not related to protein. So it's a synonymous. So it's neutral. But we know in the uncoding region, you can have some regulation role. So it has function. It's under selection. It will not be the neutral. Okay, so we need a solving this to fix those limits by go back to the relative substitution method, which I introduced a long time ago. Okay, so C is substitution rate, mu is the mutation rate. So we relative to the mutation rate, that we hope this can fix this problem. Okay, so in order for that, I have to first define the mu. What's the mu? Okay, and mu is basically random mutation rate. And so here I show example. We have the polymerase based on a template and try to make a copy of that template. That polymerase machinery is not perfect. So you're going to make a mistake. And so here we have to make some assumption about the nature of those mistakes. Okay, so we ask, can polymerase recognize position when try to copy the template? What's your idea? Can they recognize position? Now I'm in the position 1, now I'm in the position 100, now I'm in the position billion. Or now I'm located a synonymous site. Now I located a non-synonymous site. Can polymerase know those information? Partly we think it's harder for the polymerase to know that. So then our simple assumptions say those mutation rates should not depend on the position, should be position independent. They all make some similar error rate. So this is one of the assumptions about the random mutation. Second, and suppose this polymerase structure are stable, so we make some the small mutation, we assume its performance will not dramatic change. So then the error rate, copy error rate, will somehow can be, in short time at least, time independent. So that's two fundamental assumptions typically people make for the mutation step, and we follow those kind of assumptions. So with that mutation step, and here give the very simple example, you have to see the 3 strand of the very initial. So we make the copy and generate the 12 copy, and during the make copy, it's making random mutation. And because we assume the mutation will not depend on position, so you see 3 red at the beginning of the genome, and 3 green in the middle, and then 3 blue in the end, because it's not able to differentiate those positions, so it's making the same error. And then if under the neutral selection, then the substitution rate or observed from the population should be same as the mutation rate. Okay, so then the other two cases is the fixing is a high rate, then it's the adoptive mutation, and if purified, then CO mu is 1. Okay, so now with that simple thing, we can link back the CO mu with the KKS, we're familiar with it. And I only had a few minutes, and I just say to mapping back, it's straightforward. It's two conservation. Really, is the substitution rate without knowing it's a synonymous, non-synonymous should be weighted average of we know a side is synonymous side or we know a side is non-synonymous side. Okay, so with this equation, and now we can have the two simple case. Now we understand. The first case is in the translated region, and we basically can derive our familiar KKS ratio is equivalent to the CO mu under the assumption the synonymous side are neutral. Okay, we recover that facts when you use KKS, right? And then in other case, is in the UTR, which basically literally is no non-synonymous mutation. So you literally CO mu is just equal to the KS over the mu. Okay, so that's the general equation. So with this equation, then we assess the SARS-CoV-2 as the example. And so the different segment, do I have the time? Okay, so let me just go to the very end. Okay, so really my talk is that for familiar KKS analysis, now we go back to its general original equation. It's really the substitution rate compared with the mutation rate. Mutation rate is our reference state because at the mutation steps, there are no selection at all. Okay, so by this way, we have the general measure for really to determine the selection pressure. So this is the equation and we present. So we hope this equation can help to really to resolve neutralist selection debate in the long time when we apply this to analyze genomic data. And that's all, thank you.